So we're going to be taking a quick look at incomplete and codominance. So previously what we've been looking at is a simple version of inheritance, looking at just purely dominant and recessive alleles which can produce either completely dominant or recessive phenotypes. Now, these cases do exist, right? This isn't an instance of, this is how it works simply, but it's actually a whole lot more complicated. There are cases of what we've been looking at where there are traits that are that are due to one allele pair, and there is a dominant version and a recessive version, and you are either dominant or recessive in your phenotype, or homozygous or heterozygous in your genotype. Okay, that does exist. However, for some traits, it can be a bit more complicated, such as the genes or the alleles controlling the trait of colour for this cow. Now, obviously, what we've been learning so far, we would see this as being impossible, because this cow should either be completely red or completely white. However, obviously here there are places where it is purely red, and there are other places where it is purely white. In another case, we have these flowers. Just so you know, these flowers over here are the parent flowers. Let me do that. They're the parent flowers, and this one over here is the offspring. And so this offspring kind of looks like a bit of a blend or a mix of its parents' traits. Okay, you've got partly purple, partly white, and it is pink. So we have some cases where it's a bit more complicated than what we've just been looking at. So this idea of a blended inheritance does exist for some traits, okay, or for some traits in some animals. This isn't a more complicated version, than our dominant recessive inheritance. This is just a separate thing that does occur for some other traits. Okay, so we have two types of inheritance that we need to worry about. Let me just give me a pointer. Okay, so with incomplete dominance, this is a case where, there are two versions of this blended inheritance. So we have one which is called incomplete dominance. In this case, neither trait fully dominates the other. Okay, it's incomplete, so it doesn't fully get to express itself. All right, this results in this kind of a blending of the traits. So we have a mix of both, where it's kind of in between both of the traits. All right, that's incomplete dominance. The other is codominance. In this case, both traits get to dominate. Right? Like when you're cooperating, you both get to express yourselves. If you're cooperating with another person, I should say, you both get to express yourselves fully. It's just that you don't fully express yourself for everything. So we have places where, for example, in here, the white is fully expressed. Okay, that allele is fully expressed. We also have places where the pink or the red trait is fully expressed. Okay, and this is called codominance. Simply put, there is a separate phenotype for the heterozygous genotype. Okay, previously what we've been looking at, the heterozygous genotype is just the dominant phenotype. Okay, there is no heterozygous phenotype for there is no unique phenotype for a heterozygous individual. But here, with codominance and incomplete dominance, there is a unique heterozygous phenotype. So we're going to look at incomplete dominance first. The rules here at the top, these apply to both incomplete and codominance. So to avoid confusion, both, depending on how you look at it, this could be adding to confusion, but the trait, because these are both dominant traits, we will still give the trait a capital letter. Okay? Now the various alleles, however, we will assign a separate letter, also in capitals, but each allele will have its own letter. Okay, so A is for colour. Okay, the trait colour is assigned by A in this case. And the B and C are the alternate alleles. Okay, please take note that you do use different letters here. So in this case of snapdragons, which is this plant over to the right, the trait is colour. I can denote it with a capital C because these are both dominant alleles. And the two variant alleles are the red, so that's a CR, C capital R, and white, which is C capital W. Alright, so I can have these three versions of the possible genotypes. Okay, so this one over here will result in a red coloured snapdragon. This one over to the in the middle will be a white, which I'm going to use black for. And over here on the right, because we're looking at incomplete dominance, this is going to be somewhere in between. This is going to be a pink phenotype. So just as an example, let's do the cross of a red snapdragon and a pink snapdragon. So, first of all, we have CR is red and CW is white. Everything is capitals here. That is horribly messy, I'm sorry. Let's do a simple Punnett square. I'll need to make that bigger. Okay, so we have a red snapdragon. Because it's red, we know it has to be homozygous for the red allele. A pink snapdragon is going to be heterozygous with the red and a white. Okay, in this case we can definitely tell, because it's red, it has to be homozygous for the red. It can't be heterozygous like it could in our previous view of inheritance. So here we're just going to carry on like we normally do. This is going to get both red alleles. This will get one. Now, I'm not 100% sure if there is a rule saying which allele you put first in this case, because they're both dominant. So for now I'm going to put red first. Okay, so let's do our genotypes. Genotypes, what do we have? We have 2 homozygous for C red for every 2 heterozygous. And in terms of phenotypes, we're going to have 2 red and 2 pink. Okay, so these 2 down here are going to be pink, and these 2 up top are going to be red. Okay, so codominance, we use the same conventions, we write it in exactly the same way. The only thing is that we interpret the results differently. Okay, we just interpret the genotype to mean a different thing. I should just describe. In this case, we have a speckled chicken. This is the product of a black chicken and a white chicken. So the black is fully expressed here, or black or a brown, I guess. Oops. And the white chicken parent, its allele is expressed kind of here and there. Okay, so speckled. Let's cross 2 speckled chickens here. So, that mouse spot is stuck there now. That's all right. So we're going to use C. I'm just going to use B for black, and CW is white. So we're going to do this cross. Remember, we do it the exact same way, we just interpret the genotype a little differently. So we have two speckled, so each of these parents is heterozygous. Now this one, let me just carry this through, CB, 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 CW, apologies for that, CB, CW, CW, CW. Alright, so this is the exact same as you've done previously. Okay, there's nothing new here. All that happens, the only difference is going to be in our interpretations. What do I mean by that? So, genotypes, let's do that first. We have 1 homozygous black, 2 2 heterozygous to 1 homozygous white. Right? That you've done before. It's just we're using a different way of writing the allele, but it's the same type of notation for the genotype. Phenotype, though, we get to say 1 black. That's this one over here. We're going to now have 2 speckled. Okay, this is where the main difference is. All that's happening is you're interpreting this heterozygous genotype to mean something differently. In our previous look at inheritance and simple dominant recessive, you would say that this has the dominant phenotype. In incomplete dominance, this has a mixed phenotype, where it'd be somewhere halfway in between. In codominance, you could say it's speckled. You could say speckled black and white. Usually, we would define exactly what that speckled phenotype is. So you may see things like it's a tabby cat, or it's a roan cow. But in this case, it's a chicken, so it's a speckled chicken. So 1 black to 2 speckled to 1 white. Okay, and that's the offspring from the cross of 2 speckled chickens. Okay, so our blood cells actually also show some codominance. If you know anything about blood types, you would know that you have an option for an A blood type. I'll do this in a nicer colour. You could be A, you could be B, you could be O, or you could be AB. Right, so this AB is a little bit interesting because that is kind of like what we've been looking at just in the past few minutes, where we have this A allele and this B allele both being expressed. So what do these mean? Let's go through a little bit more. So this AB is actually a type of codominance. How we write our alleles for blood is it's actually with this I. Okay, I don't know why we use I, but we have I. We also have, actually let's just focus on this first. You have IA for the A blood type, and IB for the B blood type. You also have, however, a recessive blood type as well, or a recessive allele, I should say, for O. Now, what do these mean? So, this A, B, and O, what that refers to is the flags on the surface of your cells. Recall when we were looking at viruses, we were saying that each virus has markings on its surface, or it can detect the surface markers of body cells. So our blood cells all have these markings around them, like little flags. Actually, it's not just blood cells, it's all of your body cells will have a flag to tell your body that they're on the same team. It identifies it as your own body cells. So you'll notice that there is an A flag that looks like this. There is a B flag that looks more like this. Okay? And there is also the alleles to have no flags at all. And if you look at AB, you'll see that how we know it's codominance is because you will see, what don't you see? You do not see a cross between an A and a B flag. You don't see a weird colour and a weird shape. We see both the full B antigen or flag and the full A flag. Okay, that word flag is referring to antigen. Okay, that's all that means. It's like a little marker. So your group A blood type cells will have an A antigen. Your group B will have a B antigen. Your AB has both antigens, and your blood type O has no antigens. Okay, so we have 3 alleles. 2 of them are codominant. Okay, the IA and IB, capital I's, are codominant, and lowercase I for no antigen is recessive. So if you have one allele that is for the A antigen and one allele for the B antigen, your blood type is going to be, let me just write it, type AB blood. If you have one B allele and one recessive no antigen allele, we go back to our dominant recessive rules. So this will be expressed, this will be masked, so you have type B blood. And lastly, if you have two alleles for no antigen, you will not have any antigens on your blood type, on your blood cells. So you have type O blood. Those are your phenotypes. And these should all say blood at the end. Alright, so possible genotypes. If you have type AB blood, well, we know you must have one A allele and one B allele. So we can write that as I capital A and I capital B. It's the only possible genotype. For the A, for blood group A, well, we know that we can have this. We're going to be homozygous for the A allele. But remembering dominant and recessive inheritance, or dominant recessive alleles, we can also have this. We can be heterozygous for a dominant A allele and the recessive O allele, okay, or the lowercase I. And for O, because it's the recessive phenotype, we can only have the one possible combination that will result in that phenotype. Okay, that's the only genotype that will cause that phenotype. Okay, so hopefully you can do a little bit about these, a little bit of these things. We'll practice this more in class. Just before we wrap though, why is this whole thing about blood types important? Well, the whole point of flags, if you think about it in an army or for a sports team or whatever, is to identify, yes, who's on your team, but possibly more importantly, to identify who's not on your team. And how your body does that is it doesn't just recognize what's not on your team, but it prepares to kill whatever is not on your team by these antibodies. Remember, antibodies fight bacteria, usually, or germs. Viruses. Antibodies will be against viruses. And so what's happening is that your body, if you have A antigens, your body will possess antibodies for B antigens. Okay? It's recognizing and it's treating this B antigen as if it was a foreign body, such as a virus. Okay? So what happens is you are ready to fight against anything that's displaying a B antigen. If you have a blood transfusion and you accidentally receive the wrong type of blood, these antibodies are going to attack the antigens on this B cell, and it will actually cause your blood to what's called agglutinate or clump up, and you can die from that. In a similar but opposite way, if you are blood type B, you will possess antibodies against antigen A. And so if you receive a blood transfusion from someone with type A blood, your antibodies will fight that, cause it to agglutinate and clump, and you will be in a lot of trouble. If you are group AB, however, both of these flags are friendly. You do not want to be attacking anything with those flags because those are your own body cells. So you do not possess any antibodies to those blood types, I should say. You do possess other antibodies. But what that means is you are able to receive both A blood and B blood and be perfectly fine. Okay, I'm going to do that in green because that's the color of goodness. So you will be perfectly fine. Okay, you can receive, so we call these guys universal acceptors because they're in a lot of luck. They can just take blood from any blood type, even blood type O, I should say. Okay, because blood type O doesn't display any antigens, which doesn't matter because you have no antibodies. Blood type O, however, is not in such luck. These guys don't have any flags, which means that they are ready to fight against any cell that is displaying either of these two flags. So if they receive blood from someone with type AB, type B, or type A blood, I'll put a cross here because we don't want that, they will have that agglutinating clumping reaction and they will be in a lot of trouble. However, because their blood doesn't contain any antigens, they can actually freely give blood to any of these. I should do this in green again. They can give blood to anyone regardless of their blood type. They can give to type O, they can give to type AB, they can give to type B or type A, because regardless of what antibodies are here, they're not going to detect anything on the surface of this cell. So these guys, blood type O, are called universal donors. And it's actually really important for people with blood type O, first of all, because it's universal, it can be donated and received by anyone, but it's also because O is the recessive genotype. Okay, sorry, it's the recessive phenotype caused by the recessive alleles. So there is a lower percentage of people with blood type O in the population. So it just so happens that the most useful blood to have, if you're a blood bank, is also the rarest. So, and my computer's just died. So the people with the rarest blood type O really have this extra necessity, or this extra pressure to donate blood, because their blood is so useful. And that's going to wrap what we're talking about today.